All right, I think we can go ahead and get started. So on behalf of Biocytogen, I'd like to welcome everyone to today's webinar. We're very excited to invite back some of our antibody discovery experts that will provide some insights into the capabilities of our RenMab, RenLite, and RenNano platforms here at Biocytogen, so we can help you achieve your research goals. So before we continue, I'll briefly introduce myself. I'm Jenna Frame from the marketing team here at Biocytogen. My scientific expertise spans the use of animal models in the fields of hematology, oncology, and immunology. I'm joined today by our CEO of Biocytogen Boston, Dr. Jinkong Lin, who's an expert in antibody drug discovery, and he'll join the Q&A panel following today's presentation. And finally, I'd like to introduce today's speaker, our scientific director, Dr. Li Hui, who has over a decade of experience in antibody discovery and has been leading the antibody discovery team here at Biocytogen for just about 3 years. So without further delay, I'd like to invite Li to begin her presentation.
Okay, everybody can hear me? Thanks for the nice introduction, Jenna, and hello, everyone. Thank you for attending our webinar today. I will give you an introduction about our human antibody mouse models, RenMab families today. So this figure shows the current major approaches that are used for therapeutic antibody discovery and development. The first one is immunization of wild-type mice. Let me get the laser. Is immunization of wild-type mice using hybridoma technology to discover and develop mouse monoclonal antibodies, which go through humanization process to become humanized antibodies. And the second is phage display library, using human antibody library. Through design of the library and multiple rounds of biopanning, the human antibodies are obtained at the completion of the process. The third one is using transgenic mouse models, which I will describe in detail today. These mice carry human immunoglobulin gene. Immunization using these transgenic mouse generates fully human antibodies in vivo. The fourth one is obtaining human antibodies from patient PBMCs. Best example of this is using convalescent COVID-19 patients PBMC to obtain neutralizing antibodies for SARS-CoV-2. So at Biocytogen, we provide transgenic mouse models, which is a RenMice platform, and we also provide PBMC antibody discovery platform in our company. So for researchers who prefer to start with the wild type mouse antibody, we also have expertise in generating wild-type mouse monoclonal antibodies and humanize them in vitro. The only difference is we use a beacon system instead of hybridoma. Here comes the question: to human or to humanize? That is the question for everyone who works on antibody discovery. How do we choose which route to move forward? As reflected by this chart shows the number of different formats of antibodies therapies approved each year. The red one is the fully human antibodies, and the teal ones are the humanized antibodies. So far, we can see that fully human antibodies have picked up speed during the last 9 years, and the humanized antibodies are still the main contributors to the approved therapeutics. Let's compare these strategies. For humanized antibodies, the advantages are the original antibodies can come from species other than a mouse and a rat. Although the development timeline is longer and the humanization process might compromise the developability of some antibodies, currently we see great advances in computational technologies, which helps increase the success chance of the humanization process. For fully human antibodies generated by transgenic mouse or rat, the unique strength is that antibodies are naturally generated, selected, and affinity matured in vivo, using the evolution fine-tuned mechanics of the mouse or rat humoral system. The fully human antibodies generated in vivo can therefore go through further downstream development without humanization and affinity maturation steps. The disadvantage is that accessibility to the human antibody mouse can be an issue, especially for academic researchers. The process of licensing a transgenic mouse model can be lengthy and complicated due to various reasons. For human antibodies generated by phage display, the advantage is that the library sequences are from human. Also, the high throughput screening and engineering capability is also very attractive. Phage display can also be very useful if the antigen has very low or no immunogenicity, or it is toxic to the host animals. The disadvantages of these platforms are some sequences are hard to be expressed in phage system. And sometimes scFv binders derived from the phage display may lose binding upon conversion into full-length IgG. And also the quality or diversity of the library can be a limiting factor of the platform. Now, please allow me to introduce our RenMice platforms. RenMice are a family of with three family members. So these RenMice families are the mice of their immunoglobulin gene humanized. So we have RenMab that carries human heavy chain and kappa light chain variable domain to enable us generate fully human antibodies in vivo. RenMice carry human heavy chain and fix the human kappa light chain. RenLite mice is designed for bispecific antibody discovery. And our newest member, RenNano, carries human heavy chain variable domain only and is designed to facilitate the discovery of nanobodies, heavy chain only antibodies, and multispecific antibodies. This is a genetic illustration of RenMab mouse. I'll walk through this slides with you slowly. The left side is heavy chain and the light side is kappa light chain. The green color is the mouse genome and the red color is the human genome. So our scientists used a proprietary chromosome engineering technology to knock out the mouse heavy chain variable domain. And in situ, they knocked in the whole 1 megabase human VDJ segment. This segment is the most comprehensive coverage of the heavy chain variable domain in the field. Since it not only includes all functional V genes, but also includes non-functional V genes, such as pseudogenes, and also include introns and cis-regulatory elements. This is what we differentiate from our peers. In another word, we have the most human-like system here. The constant domain keeps mirroring for proper B cell development. The right side is the kappa light chain. Similar as the heavy chain, the mouse kappa chain variable domain was knocked out, and in situ, we knocked in full segment of human kappa light chain, including proximal and distal V clusters. For this version of the RenMab, we didn't disrupt the mouse lambda light chain, but we also made a version with a mouse lambda chain knocked out if our partners prefer. Here is the historical summary of the first generation and the second generation of human antibody mouse platforms. This table is not complete and only contains the main players. The first generation includes OmniRat and KM mouse, Xeno mouse, and Harbor H2L2 mouse. The first generation is featured by knockout of the mouse immunoglobulin gene plus transgenic, plus introducing the human counterparts using transgenic strategies, which means the location of the human immunoglobulin gene that inserted is random, so it's not well controlled. The second generation used a knockout and knock-in strategy, which means the human counterpart is introduced in situ. So this gives better control compared to the first generation. So here, as you can see, Biocytogen's RenMice platform, we also use in situ replacement. Here's what we different, as I mentioned earlier, we have full coverage of the heavy chain and kappa light chain genes. And the other peers use different strategy. Also, they selectively knock in different numbers of functional V genes. So overall, the RenMab uniqueness are three major points. One is we have broader diversity. Two, we have more human-like repertoire based on the reason that I just mentioned. And thirdly, we have robust immune response. The most comprehensive case study for RenMab platform is SARS-CoV-2 campaign. Although this data is a bit aged, I still would like you to pay attention to the 10 best hits we identified from RenMab mice. These antibodies have high binding affinity at nanomolar, sub-nanomolar, and even picomolar range. They also bind to diversified epitope. As this figure shows, these 10 clones bind to 4 different epitopes within RBD domain. And these clones are also functional. They can block the interaction between human ACE2 and spike protein. And when combined 3 of the antibodies that recognize different epitopes, these antibodies can neutralize almost all the variants at the RBD domain. RenMab mice have robust immune response against various targets. This slide shows RenMab mice serotiter curves from 4 individual campaigns with 4 different targets. We can see in each campaign, all RenMab mice show very robust immune response against the individual targets. Our scientists also tested RenMab with the GPCR target. This slide shows the serotiter from a GPCR target campaign. Among these 10 RenMab mice that were immunized with the DNA, almost all of them show titer at 1 to 10,000 dilution, and 142 positive hits were derived from this GPCR campaign, from the following beacon screening. And out of these positive binders, 58% derived antibodies, the GPCR antibodies, bind to the live cells overexpressed with this GPCR target. That's a pretty high success rate, given the difficulty and challenge of the GPCR protein antibody campaign. So RenMab-derived antibodies also show decent developabilities. As this slide shows, the melting temperature, the HIC, HPLC, purity, and isoelectric point data shows that the raw RenMab antibody sequences without engineering show pretty decent developability. So this provides a solid basic foundation for further engineering. To summarize, RenMab mice carry most diversified human immune repertoire. It also the antibody derived from RenMab mice has very desirable developabilities. RenMab mice show robust immune response against various targets, including GPCR targets. And the binders generated by RenMab mice has high affinity and also bind to broad diversity of epitopes. So now let's move on to RenLite mouse model. RenLite mouse is the 2nd generation in the RenMice family. It is designed to facilitate the construction of bispecific antibodies. Just as this cartoon shows, if the antibody against the target A or B has same light chain, then the combining effort of these 2 antibodies to make bispecific antibody will be much easier, since we don't have to worry about heavy and light chain mispair issue anymore. Just like this figure shows, if the common light chain are different between these 2 antibodies, then there are 4 different types of combination, and we will need to fish out the right combination clone. And this will cost extra time and effort during the development process, and it is not ideal for CMC process as well. By using the common light chain, RenLite antibodies avoided this type of issue. So here is the genetic illustration of RenLite mice. The heavy chain is the same as RenMab, and I won't repeat here. The light chain modification is different. After the mouse kappa light chain variable domain is knocked out, we knocked in one single human kappa V gene and one human J gene. This way, the kappa light chain remains fixed and the VJ recombination is avoided. So one thing I need to emphasize is that this RenLite mice, since it has the same heavy chain variable domain as the RenMab, that means RenLite mice still carries the highly diversified full coverage of human heavy chain immune repertoire. Here's a case study. We immunized RenLite mice with TFR1 protein, which is also named CD71. Here, the title curve shows a very robust immune response from RenLite mice. After 3 immunization, we can get EC50 of 400,000 from the best mouse. We use the beacon for our antibody discovery process. For this TFR1 project, we identified more than 2,000 positive B cells in one screening. This is a sample view from the beacon chip, and look how beautiful these blooms are. The beacon system allows multiple assays in one screen. Other than specificity of antigen binding, we also performed the in-pen competition assay and identified hundreds of TFR1 antibodies that have competitively binding with transferring and the ones that do not compete with the transferring. So here's some sample curve of the competitive binders. With one common light chain, one might think that epitope binding might be affected. However, we still observe pretty diversified epitope binding of RenLite-derived antibodies. As this figure shows, 15 RenLite antibodies were classified into 4 different epitope bins, which contains total 9 different epitopes. Here's the developability of 25 RenLite antibodies from one particular campaign. This table shows that almost all the antibodies have very good expression yield, high purity, solid thermal stability, and hydrophobicity and isoelectric point. Here are some SMIC assay results. SMIC is stand-up monolayer adsorption chromatography. These assays reflect the non-specificity binding and precipitate and aggregate tendency of the antibody molecule. But this data shows that RenMab, both RenMab and RenLite mice have very low retention time, which is very comparable with 95 FDA-approved IgG1 antibodies. Here's another case study. I have presented this data before. TIGIT antibody have gone through multiple pitfalls, but with recent encouraging news from Roche's tiragolumab phase 1b/2 trial for advanced hepatocellular carcinoma, it seems necessary to present it again. The quick introduction: TIGIT is one of the inhibitory immune checkpoint protein, and it's a hot target for biotherapeutics development. Currently, there are several therapeutic antibodies in clinical trial, but none has been approved by FDA yet. So we had a TIGIT discovery campaign to compare the RenMab and RenLite mice head-to-head. 5 RenMab and 5 RenLite mice were immunized with recombinant human TIGIT extracellular domain protein. And here's the titer from both groups of the mice. The immune response is pretty robust, as you can see. The beacon screening obtained 271 positive B cells from RenMab mice and 225 cells from RenLite mice. And the best clones show pretty strong antigen binding affinity, which ranges from double-digit nanomolar to picomolar. Most of the hits can bind to TIGIT competitively with its main ligand CD155. So the red one is competitive binders and the white one is non-competitive binders. Based on this data, we learned that RenLite mice show robust immune response and yield a high number of hits against multiple targets. And RenLite mice can generate high affinity antibodies that bind to diversified epitopes. RenLite mice generate antibodies that have solid developability. So overall, it is a great platform for bispecific and multispecific antibody discovery. Next, let's talk about RenNano. Third family member RenNano is the newest member of the RenMice family. As this genetic illustration shows, heavy chain variable domain is the same as RenMab. The constant domain was modified to ensure only heavy chain antibody is generated by the RenNano mouse. The light chain has been knocked out. Please keep in mind that since the heavy chain variable domain is still the same as RenMab, that means our RenNano mouse also have the very diversified, comprehensive coverage of all the functional V genes and non-functional V genes. So here are the peers in the market. We can see that RenNano mice, compared to Harbor HCAB mouse and UniRat and Crescendo mouse, our RenNano mouse differentiate from the peers that we use in situ replacement, while the other models use transgenic insertion. And we have the most comprehensive V gene coverage compared to partial coverage. And so this is what our uniqueness are. Here are the example of RenNano serotiter from 3 different campaigns. We can see that the titer is pretty decent for mouse that generate only heavy chain antibodies. These are 3 different target campaigns. Again, we performed the RenNano campaign for TFR1. TFR1 is one of the main cell surface receptors that can mediate transcytosis across the blood-brain barrier. We also call it BBB. So a nanobody or heavy chain antibody against TFR1 will be a useful tool to carry payload across the BBB. So as the workflow shows, 8 RenNano mice were immunized and moved forward to Beacon after titer reached plateau. Beacon screening obtained 268 positive B cells, and among these hits, 63 human cyno cross-reactive antibodies were identified by flow cytometry. And among these 63 antibodies, there are 31 have good binding affinity with both human and cyno TFR1, tested by SPR. Then internalization assay was performed. 7 clones were shown to have positive internalization. Then a few of these clones were moved forward with in vivo crossing BBB assay. This data shows the RenNano-derived heavy chain antibody binds to cells overexpressing TFR1. We can see the majority of the hits are positive. Here's the heavy chain germline usage for these 63 clones. We see IGHV family of 3, 4, 1, 5, and 6. It is pretty diversified. And CDR3 length also shows that the majority of the antibodies have a CDR3 longer than the 14 amino acid. Here's the internalization data for the 7 clones and their binding affinity. We can see that with heavy chain only, RenNano antibodies still have pretty high binding affinity, all at the nanomolar or double-digit nanomolar range. And here's the internalization data. Compared to the positive control, these antibodies can be internalized fairly easily, but you don't want to have too strong internalization, because sometimes if internalization is too strong, that means the antibody might be trapped within the BBB and not go through it. So we would like to choose the antibodies that have decent internalization, but not too strong. So our scientists inject 3 antibodies, RenNano antibodies that show good internalization in data into the vein of the mice, and then monitor the level of TFR1 heavy chain antibody in blood as well as in the brain after the saline perfusion. TFR1 heavy chain antibodies show higher concentration in the whole brain and parenchyma after injection compared to the positive control. We can see 1 clone antibody AB38 is the red one, which shows significantly higher brain concentration or brain distribution amount compared to the positive control. And also these concentration in the brains also depend upon the dose. It's dose dependent. And then our scientists tested stained the brain tissue using the secondary antibody and then the neural marker. And you can see that the antibody 38 at the medium dose can show a very good staining, positive staining in the cerebrum and cerebellum, which also stain the neuron and other tissues in the brain as well. So these TFR1 antibodies also have pretty high binding affinity around nanomolar range and solid developabilities. Here we show more detailed information about the developability from the three best RenNano heavy chain antibodies. These clones showed pretty high purity, short retention time in HIC-HPLC and CIC-HPLC, which indicates superb hydrophilicity and specificity. And also the SMAC time, as I mentioned earlier, and also favorable thermal stability. We will have more developability data coming out soon in the near future. Please stay tuned. This is another case study. 4-1BB campaign using RenNano mice. Left side is the titer. The right side is the derived 4-1BB heavy chain antibodies that bind to the overexpression cell lines with 4-1BB. You can see that the majority of the hits bind to the overexpressed cells strongly. The selected hits also have in vitro agonistic activity as shown here in the reported assay. When 4-1BB overexpression cells were co-cultured with Fc gamma receptor expressing cells and treated with the antibody, you can see that the RenNano 4-1BB antibodies exert very strong in vitro agonistic activity, functional activity. To summarize, RenNano mice carry diversified and comprehensive human heavy chain repertoire. It have decent immune response against various targets. RenNano mice generate functional heavy chain antibodies with solid developability. Overall, it is an ideal tool for generating heavy chain antibodies and nanobodies. To wrap up, today I introduced to you our RenMice family. The RenMab platform provides full human heavy chain and kappa light chain repertoire diversity and generates high affinity and high diversity binders. The RenLite platform accelerates bispecific or multispecific antibody discovery with full heavy chain diversity and desirable developability. The RenNano platform is an ideal tool for heavy chain antibody and nanobody discovery with full human heavy chain diversity and solid developability. So currently, we have established partnership with multiple big pharmaceutical companies and biotech companies, including Merck, EMD Serono, Zenker, Janssen, and Beijing, and we are in communication with more companies as we speak. Please contact us if you are interested in partnering with us on the RenMice platforms. We have a great antibody discovery service platform that can support all RenMice antibody campaigns. And thank you for your attention. Please feel free to ask questions.
Thank you so much, Lee, for the fantastic and informative presentation. Looks like we have at least one question coming in so far. Hopefully there'll be more. So how do you approach immunization and what types of immunization strategies do you offer?
It's a good question. So basically the RenMice platform, that the uniqueness and the strength and also what we differentiate from other peers in the market is that we have robust immune response. So basically for RenMab and RenLite, we can just immunize the mice as wild type mice because it is expected that the RenMice will have very similar immune response towards the antigen, very comparable to the wild type mice. For RenNano, since it is heavy chain only antibody mouse, which does not have the kappa light chain, which means immune response can be compromised to some degree, but we still can reach very decent titer with extra boost or two.
Adding on to Li's answer, I think more important is for the challenging target, including GPCR, is challenging for wild type as well. Then Li and our antibody team also offer DNA immunization and other immunization strategy, including the virus particle as well.
Thank you, Dr. Li. Yes. Yeah, we also offer a DNA immunization, cell immunization, VLP immunization, like Dr. Li mentioned. So for different targets, we can always customize based on the challenge and the nature and biology of the target, and then tailor the immunization strategy for you.
Okay. Okay, great answer. All right, next question. Is there any lead time for a campaign? How long would a typical campaign take?
Yes, so the lead time for initiating any new campaign is about 2 weeks. The typical campaign normally lasts from 3.5 months to 4.5 months, depending the challenginess of the target. Some target needs to lengthen the immunization if the immunogenicity is relatively lower, and also depending on the how many screening strategy and the late stage validation steps are needed. So that can be variable, but overall it's between 3.5 months to like 5 months, like the longest.
Okay, and that would be for any RenMab, RenLite, or RenNano strain, similar.
Yes.
Okay, similar timing. Okay.
For sure, we need to reserve these mice from and shipping over from China for your study.
Yeah, that takes about 2 weeks.
Okay. So is the immunization only done by Biocytogen? Are there certified providers available?
Yeah, so we have our vivarium available here at Boston, and we can take care of the immunization. However, if our partners prefer to use a third-party certified CRO company, we can cooperate with that as well.
Great. Next question. So what kind of licensing options do you have for academic institutions to collaborate?
Yeah, that's a good question. Dr. Lin, do you have any comment?
Gina, what's your question?
So the question was specific for academic institutions. What kind of licensing options would we have for academic institutions to collaborate?
Yeah. So we are very open. At the moment, we do a lot of co-development and co-research with the academic. I think we are not going to use the standard industry licensing to deal with the academic professors.
Okay, thank you. And yeah, definitely reach out to us afterward for more discussion. So next question is, how many mutations in the framework of the nanobodies are obtained on average?
That's a very good question, a great question. It's also a tricky question because we have observed some mutations in the framework of the nanobody, because nanobody doesn't have the light chain, right? So the heavy chain light chain contact area normally would need a charge change to change the hydrophobicity. We do observe like a portion of the antibodies would have the automatic mutation in the framework 2 and framework 4 area to increase developability of the particular RenNano antibody.
Okay. So next question is regarding the TFR1 campaign. So do we license out TFR1 antibody leads using the RenNano platform?
Yeah, we're welcome. We're open-minded. So if you're interested in our RenNano, we can connect you with our team, and then we can discuss further about it.
Mm-hmm. Yep, we have a large number of assets with a separate licensing team that would be ready and willing to speak with you. Okay, next question. Could you please compare RenMab versus Alevimab mouse platform?
That's a good question. Yes, good question. So Alevimab, they use, I think, first of all, they did the strategy, they knock in the mice is different. I think they either use BAC or YAC, I don't remember exactly in detail, but our proprietary chromosome engineering technology is very unique. So this is what we're different from them. The second thing is the Alevimab, I believe, if I remember correctly, they knock in selective functional V genes with a limited number of V genes. And we knock in the whole segment, which include everything, like I mentioned earlier. And I think also the Alevimab, if I remember correctly, they use artificial way to synthesize different genes and put them together. And, you know, again, our model, RenMab, is the natural whole segment flip in. So I think if Dr. Ling, if you have anything to add.
A good question. And be honest with you, we are not allowed to do head-to-head directed lab comparison. All of these mice are confidential. And particularly for Alevimab, they never disclose their detail of the strategy like what we did. Like what Dr. Lee just said, we do the chromatin engineering, do the entire cassette. VDJ knockout, knocking, VJ knocking, knockout, and also including all the pseudogene, intron, exons, all of these regulatory elements. If you look into the antibody low site, you'll see a lot of hypersensitive regulatory elements will be in intron, will be included in our mice. Therefore, our mice behave like wild type. So bottom line, RenMab behave like wild type, nobody else, including Alevimab, are comparable to wild type. That is what I can say. Thank you.
Okay. Thank you, Dr. Lin. So next question is, do we have capabilities to test the in vitro and in vivo efficacy of antibody candidates?
I'm sorry, can you say that again? I was clicking.
No problem. Okay, so the question is whether we have the capabilities to test in vitro and in vivo efficacy of the antibodies.
Oh, yes, definitely. So obviously, our company has a great pharmacology platform, service platform, that we have tons of humanized target mouse models for in vivo efficacy testing. And we also have in vitro team as well to accommodate all the in vitro efficacy testing as well.
Okay. All right, next question. Are there differences in CDR3 length between RenMab and RenNano?
So the RenNano seems to have longer. I mean, based on, so obviously the data is still limited so far. We don't have, we're still generating tons of data as we speak. So the pool is very small, but so far we have observed that the overall CDR3 length is longer in RenNano than RenMab, which is understandable, right? Because there's no light chain there. So the heavy chain that with longer CDR3 would be more better selectively selected in vivo to survive.
So direct answer also is like RenMab, we have about 14 instead of the median length. And then RenNano, we have a little bit longer, so 15 to 16 amino acid.
Thank you, Dr. Lee.
Okay, next question. Looks like maybe a contractual question. How does, with murky IP landscape, how does Biocytogen address the freedom to operate concern of a partner?
I'll leave that to Dr. Ling to answer.
Any question? Any comments?
This is legal question, very nice legal question. My answer, and we do, as a scientist, we do have the free operation for the legal aspect. In terms of IP, I think that is a question for our legal team to answer. We scientifically, be honest with you, we use very unique special technology to do these models. So as a scientist, I believe Biocytogen have our own IP. And we have the freedom to operate, for sure. That is for sure.
Okay. Another question about RenNano. So could we compare RenNano versus AAV-mediated nanobody delivery to the brain? For example, toxicology or efficiency.
That answer I'm not 100% sure because we haven't done AAV mediated delivery to the brain yet.
I think it's different strategies. There are no direct comparison to include over here. RenNano, because of small binder, therefore you have the certain levels of penetration, but in terms of toxicity, it's totally different from virus delivery system toxicity. There's two different methods. It's not direct comparison regarding the toxicity or even efficiency.
But this is a good question. I mean, in the future, I think our team should try to compare them to when we try to figure out the best therapeutic strategy.
Yeah, thank you for these insights. Okay, so next question. Do we perform the RenMab platform, or maybe we're referring to RenMab as a whole? Do we perform these projects in USA or in China? That I can answer, and I can say that we have a large team in China with a huge animal facility, but we also do have a team here in the Boston area. So if you are local to Boston or on the stateside, our team here will be able to support your needs. Anything you want to add?
Thank you, Jenna. We have a great antibody discovery team here. We have a beautiful beacon machine here. So everything can be done here locally, and you can drop off your sample. We can deliver your sample. It's very convenient if you want to perform your antibody campaign locally with us in here within the US.
In addition to that one, we also can do the antibody campaign in their own facility in case they want to do this different strategy. So mice also are open now to really work closely to you, depends on your needs.
Yeah, absolutely. Thanks for adding that. Okay. And
no more questions.
No more questions. So, looks like we have another one just popped in. Is it fair to say that the RenMab platform can deliver more diverse hits than some of the CRO partners that are using machine learning or AI?
We need to think about that. Because now with the machine learning and AI, it's there's this bright future, but we don't know yet that whether AI can outperform the RenMab platform. Right now, I still think the RenMab platform is the best, can deliver diverse hits generated in vivo, right? Using the evolution evolved this immune system. In the future, we don't know, but for now, I think RenMab still can outperform the AI.
Anything you want to add? No?
You want me to add? Comments on? It's challenging, but for sure, answer is fair. I mean, RenMab is like we have wild type of full human antibody repertoire.
Right.
And no matter AI, it still depends on our human antibody repertoire to generate a library. And cannot be bigger than what we have in our human repertoire. That is for sure. No matter how imagine, you are larger than our human repertoire, it's not truly, that will be artificial.
Right. So there's a difference between diversity and functionality.
Right. So I think we already get into the human antibody repertoire limit, right? And in vivo maturation in mice is unlimited. So you cannot calculate by 10 to 9 or 10 to 12 or 10 to 13, you cannot. So whatever we have in human, we already move into the mice.
Okay. Thank you, Dr. Lin. Okay, so it looks like for the moment, we have no remaining questions. So, while we wrap up here, I'd just like to say thank you again to Dr. Lee and Dr. Lin for joining us for such an informative presentation and panel discussion. So if there are any more questions that may come up after we close the webinar at a later time, you can feel free to contact us at info@biocytogen.com, and we'll be happy to speak further with you about your research goals. You can also head over to our website, biocytogen.com or renmab.com, for more details about these and other models and services we offer for your preclinical studies. So with that, thank you so much for joining us, and have a great rest of your day. Thank you.
Tina. Thank you, Lee.
Thank you.
everyone.
Thank you, everyone.